Hello, potential viewers. Stream will be starting shortly. Please keep your arms and legs and head inside the ride at all times. Oh, hello. Right now, we're looking at a diatom. This one is from a site in, I think, Nebraska. It's about 6 million years old or something like that. And from fossil deposits. And I was going to have some guests on today. My former student Joe and a taxonomy friend of mine, colleague, Dr. Mark Edlund. But Mark got sick. I'm not sure exactly what the problem is, but he didn't feel very well today. And so simultaneously, Joe got a option to get vaccinated, and his number got called. So he's doing the vaccination, and Mark's not feeling well. So I just went ahead and put on some of the material from the stuff that we were going to look at. I figured I don't need them. I can take pictures anyway. But yeah, so this is some material that we've been looking at previously. Avian Bob asks, how do I determine the age of the diatom? In these cases, these are ashfall deposits, or deposits where volcanic eruptions have occurred, and they used geochemical tools to date the actual eruption events. So normally they can trace the volcanic activity, the eruption that's associated with the ash, back to a specific event, and then as long as they can figure out chemically, like fingerprint, which one of those eruption events it was, then they can use that. Usually it's argon-argon dating, or sometimes potassium-argon dating, on the basalt, which is the flow that came out from the volcano. And so there's the ash that's at the site, and they know that there's an eruption that came from that volcano because the ash and the basalt should have similar chemistry. And what they do is use these geochemical techniques to actually date the basalt, which is then tied to the ash, and then the ash is the same interval where the diatoms are collected in this case. So we don't actually date the diatoms, we date the sediments that are associated with the materials where the diatoms were found. And so these are well-dated, well-known ages for the samples based on some previous volcanic eruptions. And independent of all that, basically, somebody sent me these samples. So we're just looking at the diatom component right now. But when I scroll out, when this image is done collecting, I'll scroll out and we can actually look at the ash itself, which is the thing that was connected to the volcanic eruption. In this case, we're just looking at the diatoms, but the deposits have to be the same age as the ash where they occur, just by deduction. So it's a good question. I have some other samples that are on the other stubs. So if you look at the chamber view that's below us, there's 6 stubs on there. 2 of them are from this site, which is the Grove deposit. I think there used to be a lake at the site called Grove Lake. And so this is some of that material. And there's also some material from Ashfall State Park, which is nearby. So so this is the diatom we were looking at. Diatoms are a type of algae, and their skeleton, their cell wall is silicified. And when the diatom dies, the silicified scale wall or skeleton is preserved. And you can see down here is a scale bar. So there's always an idea as I scroll in and out, you should be able to see how big our sample is. This one, if we were to have the whole thing in our field of view right here, is about 19 microns. So there are 1,000 microns in a millimeter. And of that 1,000, here are 19 of them, basically, that are the diameter of this diatom. So it's very small. And these little pieces that you can see around the outside edge are also silica. So the skeleton of the diatom is silica, but the ash is also mostly silica. And so similar processing techniques can allow us to get the material out. So all of these pieces that you see that are non-geometric shapes, they're just sort of, like random shapes, are not diatoms. And this round thing is a diatom. There's another round thing up here, also a diatom. These are internal views of the diatom. This one's got a little bit of junk on the inside of it, but you can see it's rounded. And it's got another piece of a fragment of a diatom inside of that. So it's kind of obscuring or occluding our view of the rest of it. So the age is largely determined in this case by other people. I don't actually do any of the dating, and I'm just trusting that they, they used appropriate techniques to get those ages. I think the sites are fairly well known, so they were dated quite a while ago. These are just debris again. There's also bits of sand and silt mixed in with the sample, because these are fossil deposits. And so you'll see some of that as well. But a lot of this is actually ash. So here's another diatom up here, and another one over here. And the littler ones are kind of interesting to me because we haven't seen a good internal view of a little one, or external view. Here's a nice external view. I can tell it's the outside because it's basically cupped away from us. Normally when it's cupped towards us, the diatoms have a very bright, much brighter than this margin around the outside, and that's because the outside cup of the diatom is pointed towards the sensor. And in the SEM, that's how it that's how you can tell which direction something is. It's in three-dimensional shape, and there's artificial shadows that are created by the topography of the surface, because there's a sensor inside the chamber view down here. You can sort of see there's a little grill-shaped thing. That's the actual sensor, and it's off to one side. So it creates the sense of a shadow. And I'm going to just slow this down a little. And then I'm going to adjust for the brightness, because this top part's a little bright. And then hopefully the computer will balance that out. Hey, how's it going, Luke? There was a little piece bitten out of it, yeah. How much do diatoms weigh? Diatoms are actually very lightweight relative to most other things that you might find. Their skeletons are, they've got little tiny holes in them. So I don't have an exact answer for you with respect to how much they weigh, but I can tell you, if you had a little piece of sediment that was collected from diatomite, and you weighed it against normal sediment or a rock of similar volume, that the diatom part would be substantially lighter. They are very light, lacy structures, and as a result, they tend to be pretty lightweight. So in a normal sample, like a lake sediment sample, for example, that we look at, we might have 1 million diatoms in 1/10 of a gram. So an individual diatom is very, very light. And that also includes other types of sediments, so it's not an exact measurement. It's an interesting question to think about, though. I think that's looking good. So I changed the speed settings as we image things. I have to have it on a faster speed or a lower number in the SEM in order to move around and have it redraw it quickly enough for us to actually see what we're moving to. And then I always slow it down so that we can actually see the details. So I could potentially slow it down even farther if we wanted to see the image at a slightly higher resolution. You can't really tell a difference between where I switched it and where I didn't. It's just slightly higher resolution at this point. Sort of diminishing returns after the speed that we had it at. So I usually leave it on this level unless I turn down the beam intensity to a slightly smaller size, and then I usually will slow it down so it doesn't look quite so grainy. So I have it on speed 7, which is basically the 3-minute images that we normally collect on the stream. If I had 1 liter worth of diatoms, how much would it weigh? Yeah, I don't really know. I've never had a liter of diatoms, but you could go to the hardware store and buy diatomaceous earth in a powdered form, and you could weigh out a liter pretty easily. So I could get that answer for you, and I think we actually have diatomaceous earth at home in a bag that we got from the hardware store, because people use it to sprinkle in their garden, keep slugs away, and that sort of thing. The density for most diatoms is distributed a little bit differently. So you can see even in these images, there's lighter parts and darker parts. And the darker parts mean that that's a hole or in a shadow. And you can see there's a whole bunch of shadows in here. So the parts that have brighter components on a similar elevation, so here, those have higher concentrations of silica in them because they don't have the little holes. So necessarily they're thicker. And these structures on this diatom, this is a cyclotella, or it's kind of like a cyclotella. It's not quite right. So these are the striae, or the striated component, and this is usually referred to as a costi, or a rib. And the rib components usually are, like our ribs, a little bit denser. So, and then you can see there's little patches here where there were spines. The spines are now broken off. So here would have been a spine at the end of each one of these. And they also have a little bit of extra silica. And then this margin around the outside is solid or hyaline. And as a result, it's where a lot more silica is located. And then you can see there's a colliculate part in the middle here, which would also be a little bit more heavily silicified, although it does have holes. Are they denser than water? Oh, so their density is well known. I think it's the specific density is something like 2.32 or something like that. And yeah, the middle looks like a disaster because it's got a bunch of little diatom pieces and fragments of ash and other things on it. So normally they would not have this junk on it. And I went ahead and took the picture anyway because I want some external views, and I don't know how many good views we have in these samples. So I'm just going to call this one Cyclotella species 2, even though I'm actually pretty sure it's not Cyclotella. And also, I know it's not the other species we were looking at, or it's probably not. So I don't know what species it is. It may not be described. So when you look at 6 million year old diatoms, even when you look at modern diatoms, there's a fairly high probability that what you're looking at might not be described. I mean, maybe 10% chance or something like that you're looking at something that might be new. So when you go back in time, there's very few people who've looked at even these deposits very carefully, and they probably just called it Cyclotella. But I suspect it's not a Cyclotella. It's just close to a Cyclotella. In any case. So I'm going to zoom out. We've got enough of that. And you can see, again, some large fragments on the screen. And also, I'm pretty sure we saw, there's another Cyclotella over here. Cyclotelloid, I suppose, would be more appropriate. And this is an internal view. You can see these little structures are called alveolar chambers. And there's alveolar chambers all the way around the outside of this one. This is the same as the species that we started with. It's the same organism, I'm pretty sure. And then there's some structures around the outside edge. So here's the costi structures. In these ones, the costi are a little shorter than in the previous one. The surface also has a bit of an undulation on it. And then there's some structures that you will be able to see in a second. I'll zoom in on one of them, maybe up here, so we can see them a little bit better. I'll try to get it into focus. There we go. So these little structures are Mantophotoportula. That's just a fancy name for, they're on the mantle or the margin. So if you think of like a fireplace has a mantle, or I don't know, a lake has a mantle, which is the outside edge. And then photoportula portula just means it's a hole. And photoportula is our terminology in diatom language for this particular type of hole, which has satellite pores around it. So I sometimes will rattle out the terminology without explaining it very well. But there's a hole in the center of the valve, and then there's three little slit-like holes around the edge. You can see them here and here on either side of them, these little tiny slits. They don't go all the way around. And then the structure has sort of like a tube-like thing that's coming out of the middle, which is the photoportula structure, the central tube. And on the outside of the valve, sometimes those central tubes get extended and look like spines. There are also Rimaportula structures. So right here, there's a Rimaportula structure, which means portula, again, just means it's a hole that goes through the cell wall. And these ones look like little lips. So that's where the name Rimaportula comes from. Sort of mouth-shaped or lip-shaped structures is sometimes called labiate processes. So, and you can see internally the costi or the thickened parts go longer into the center. And then also you can see a little bit of the structure of the areolae from the outside of the valve coming through on the margins. So I'm going to make an adjustment for brightness for this. I've slowed down the image. I've got it in focus. And then as you can see, it's getting a little bit brighter as it crosses that boundary, which will give us a little bit more detail on the structure and surface in here. And then I'm just going to go ahead and take that picture, and I can hop back over and see what's going on in chat. Let's see. Are there diatoms deep down in the ocean? There are diatom skeletons deep in the ocean because they died at the surface and they rained down cadavers to the bottom of the ocean. So the skeletons are down there, but diatoms are algae, which means they have, in most cases, they have chloroplasts, and they need sunlight. So they're always going to be in areas that are sunlit. But they can die, obviously. When they perish, they fall through the water column. Sometimes before they perish, they fall through the water column. And they end up, their skeletons end up on the bottom of the ocean, and they actually constitute a pretty large fraction of the skeletal mass on the bottom of the ocean. So you don't get it, where did the holes go? So this is the structure here. You can see through this part if you look carefully. There's like a shaded area here, which is actually part of that alveolar chamber. And what you don't see is that the diatom on the outside looks kind of like a bottle cap. So this is like a lip on the bottle cap that is stretched over, like completely over, would fit over the top of the bottle lid, right? And so the structures come through on the costae here, and they would poke out on the outside edge. So they look like they're going straight out, but they intersect with a curved outside surface, and so there's an external opening. So I'll find you an example of this structure on the outside of the valve, because we're looking at the inside, and it's sort of cup-shaped, and it's cupped towards us. So that we're seeing the inside of the skeleton, but these little structures basically go through the cell wall to the outside. You can see some little holes up inside these things. So there's some in all of them. These are little holes that actually are holes on the outside of the valve. So there's like a layer of silica that goes over the entire outside surface. And then on the inside, there's like a structure of ribs and then there's another layer of silica that sort of like a webs across the inside of those costae. So you can see the costae structure continue, but there's like a chamber in these dark areas in here. And there's a chamber that still continues past this little lip structure here. So that is, on the surface, would just be covered by a bunch of little tiny holes, the areolae. And then these structures actually poke through the outside as well. So we see them as larger holes on the outside of the valve. This is Cyclotella species 1 again. And I'll see if I can find us a nice view of an outside view of a diatom. And that will probably help, because I can show you where they come out on the outside. So I'm just zooming back out. There's another little diatom here. There's something here that might be a diatom. I'm going to zoom in on this one and sort of see if there's anything. Oh yeah, so there's an outside view. And here are the little holes associated with the mantle photoportula. This is a different species, but these are the little holes. And it's the same way on the big one as it would be on this little guy. These little holes would be associated with the mantle photoportula on the inside. So this is the external expression, that's what we call that, of the mantle photoportula. So I'll zoom in on one of those here in a second. I just wanted to get, I'm over brightened. And it cuts into the detail, especially when you're looking at the really tiny stuff. If it's too much, there we go. So you can see the little holes here, 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 here. These pass through the skeleton and into the inside. And then on the inside, they have that sort of clover sort of structure where the central tube with a bunch of little holes around the outside edge. So again, the middle on this one is sort of colliculate, but it doesn't have any junk on it anymore. And you can see the costi, but they're a little bit more difficult to see from the outside because they are just structures that we're seeing through the skeleton itself in that case. So also I'm going to slow it down so we can actually see the detail. Again, these are areolae, and between the areolae here, the sort of thickened part is a costi. And so there's a number of costi that are basically between each of these sets of areoles. And then the mantle photoportula is out here on the margin of those things. Mantle photoportula are all these structures that are very clearly standing out in this view on the outside, right? This is a different species than the one that we were looking at. And I can tell that based on the nature of the diatom. But the structures that we can see, but the actual structures they have are similar. Like they have similar processes, which is why we give them a group name for the whole set of mantle photoportula. Okay, I'm going to take that picture. Actually, I might tweak the brightness one more time. So we looked at these samples before. In a previous stream, we looked at some of the Ashfall and Grove Lake samples before. But I had Mallory make some new stubs for them, in part because the other ones had a little bit of junk on them, and we thought maybe we could get some of that off by doing some ultrasonic water bath. We put the sample in, basically vibrating it using these vibration techniques inside of a water bath, and it shakes some of the junk off of the valves often. So they're all a little brittle. It's the skeleton, yeah. They're made out of silica. So can you catch diatoms mid-cell division? Yes. The holes remind you of machine guns. Well, they're not dissimilar from like the barrel outside barrels of guns, I guess, if you're gun oriented, which many people in Indiana are. But you can catch diatoms in mid-cell division. It would be very hard to tell in the scanning electron microscope that you have that, because they form one cell inside of another cell. So like the cell that's inside will be made by creating one half of the diatom facing this way and one half of the diatom facing this way on the inside. And then two valves that are already there just basically get expanded outward. And then the one that's in here basically pops off with this one, and the one that's in here pops off with that one. So the problem with that, of course, is in a scanning electron microscope, we're looking at the topography, mostly the outside of the valve or the inside of the valve. And we don't have a whole lot of views where we can see both inside and outside of a valve, which is what you would need if you wanted to see them in mid-division, because you'd have to be able to see both valves basically internalized. In a light microscope, however, it functions a lot more like X-ray vision. It lets you see through the cells, and as a result, you can often see cells forming inside of other cells in a light microscope. In the SEM, it's a little bit harder to catch them in that position. I mean, I think it's still possible that you could see that there's a valve inside. If it landed on its side and the valves were getting ready to split, you probably could see it. It would just be difficult or uncommon. What kind of microscopes are used for studying diatoms? Primarily because they're so small, you need upright transmitted light microscopes to see them. You can see them on a very high-powered stereoscope, which is like a binocular microscope or a dissecting scope, they're sometimes called, but it would have to have the absolute highest magnification, and the diatom would have to be very large. You couldn't You probably couldn't identify them to species from that view for most diatoms. I suppose there's a few exceptions. But for the most part, you need an upright transmitted light microscope. I don't have any examples of one of those in this lab, but in my other lab I could show you some. This is external. And that's pretty much it. Outside of that, you need a pretty high level of magnification. So most diatoms are analyzed at 1,000 times magnification as a minimum. And there's not usually any other level to zoom in on. Most microscopes, that's basically the highest light microscope. That's sort of the highest capacity that most of them have. So that's usually as far as people get, is 1,000 times magnification. I'm totally not sure what this is at this point. It looks like a diatom, or a piece of a diatom, but I'm not positive. There's some other junk over here that I also don't know what it is. That's not a diatom. And I'm not sure what it's doing in this sample. Some sort of random biological organism that I don't recognize. Not sure what that is. This is a diatom. Oh wait. Not sure about that now. I can't tell what this is. It's a little bright. Could be a diatom. Could also be a chrysophyte. This one's a diatom. This is Alveolophora in profile, in external view. Some fragments, and here's a piece of a diatom. It's basically been shattered or fragmented down the middle. Those are two valves from Alveolophora that are still joined together, and you can sort of see internally into them, which is actually kind of uncommon for a view. Some more Alveolophora in valve view here and here. There's a lot of really little things in these samples, which is why I'm trying to be careful about. So here's some little thing right there. That's a diatom. Here's a diatom. Pretty sure. Yeah. So you can see this sort of costy structure here and a raphe running down the middle. And if we zoom in, you can see the raphe ends very clearly. And we're at beam intensity 10, which is a little bit bright. So I'm going to drop it down to 7 so you can see a little bit more of the detail. Here. And even when this is in focus, it's a little cruddy because the sample's a little dissolved and broken and probably has some clay on it. But I think that's a gomphanema. It's not a very good view, so I'm going to skip it. I got some new stubs and some new materials that I'm using today, and actually spent a fairly large fraction of the money that we have accumulated since I started streaming to purchase this stuff. So that's nice. Some of it's here being used for the stream. Sometimes it will also be used for student research projects. So I should say this is sort of like a nice capacity, that we're looking at something that's basically currently entirely funded by our viewers. So thanks to everybody who's watched the channel over the last, I don't know, I guess we've been around for almost 8 months now. So your generous subscriptions and anything else that you've contributed to the channel is actually supporting what we're looking at right now. So it's pretty cool. And some of this will ultimately probably be published. It's going towards Joe Mohan, who's one of the students on the project, is a PhD student currently, and he's getting close to graduating. But it will actually go towards stuff that we will publish together. So probably describe some of these species. I can find some better views of them. This one's got a bunch of clay on the inside. That's a diatom. And this looks like maybe it's just a fragment of something. There's another diatom, just a fragment. So when I click with the mouse, it basically moves whatever I click on to the middle. So that got moved to there. And so we can move around in this SEM by just clicking with the mouse, which is nice. It allows me to very easily move around in the landscape of debris that we're in, and I can kind of keep track of where we are pretty well. There is a coordinate system, so if I wanted to, I could be very careful about where we have and haven't explored. But since I'm zooming in and out quite a bit, and most of the time I can kind of remember what was on previous fields of view and use that to make sure that we're still sort of lined up with where I want to be exploring. So we're primarily looking at the planktic diatoms in today's stream from these samples. And part of the reason for that is it's a little bit easier. They're a little bit better preserved, and it's a little bit easier to see stuff on them right now as a result. A monster fragment. So earlier somebody asked, how do we know how old the diatoms are? A lot of times we also will come up with a tool for figuring out how old the diatoms are just by looking at the diatoms. So if you get enough information from existing chronologies where they date ash deposits or anything else they can put a date on, a lot of times you can figure out when a species has evolved. And over really long time frames, you can use that information to actually tell you, oh, I see these things in the sample, so it's this age, because they evolved at a specific interval in Earth's history in the past, and they were around on the landscape for, in many cases, a short time. So that actually allows us to sort of pinpoint, using just the diatom species, how old something is. I don't know that we have that information perfectly determined for most of these diatoms yet, but it provides us some context for what we're seeing. So there's a diatom. This one is a Nitzia. This structure on the edge is a fibula, and there's a raphe that runs around the outside margin on this side, just on one side, on a keel. That's not a very interesting Nitzia to me. Many of them are not interesting to me, but that one also looked a little cruddy. Otherwise I would have probably stopped and take a picture of it. So for now I'm just kind of scouting around looking for diatoms and anything else we might come across. It's a possibility we could come across things like sponge spicules or chrysophytes or pollen grains. These samples had some additional processing that went on because they were mixed together with ash. And so we tried to use heavy liquid separation, which is basically floating the diatom material on heavy liquids. And we can set the heavy liquids to be specific densities, and then from there determine if it's floating on it, right? We could use that to determine what we see in the sample by excluding things that are lighter and heavier than it. So Luke asked this question earlier about densities of diatoms, and it's actually relevant to what we're looking at, in that knowing the density actually allows us, in this case, to separate some of the diatoms from the ash and try to reduce the amount of clutter in our field of view. It's imperfect because a lot of times the ash gets trapped on the same layer. It may have a similar density to diatoms, although usually ash is a little bit heavier because it's, slightly more dense material. So that direction was not very fruitful, so I'm going to try this side. And what we're looking at is obviously very, very small, which doesn't help. If we're scouting around, if I get too far out, I won't be able to actually see stuff. Like these little diatoms here probably would have been missed. This one's a little cruddy, so it doesn't help. Hey Micah, welcome back. Got some lunch? Fueling up for the day? I'm not sure what this is. Piece of something. A little bit bigger. There's another one of these Gomphanema, and also I can see that it's got junk in it. So despite the fact that we process these using ultrasonic water bath a couple of times already, there's still some stuff that's basically stuck to the outside of the diatoms in many cases. This one's just a little bit bright, but that's an Alveolophora. It's kind of like a crooked angle. I'm going to slow that down and then fix the brightness, so it's a little too bright. Hey, Quarkmark, thank you for the subscription. I had the sound off, so I can't hear anything. Your mama is so fat I can see her without an SEM. Well, as a diatom, yeah. Some of them are kind of big. What's the life cycle of a diatom like? Are there stages of development like pupation? Does the background seem really black today? Oh, in some cases it is. It's just got a lot of stuff on it. I mean, it doesn't have a lot of stuff on it. So it's making it look extra like studio-y. I'm going to go ahead and slow that down and take that picture so I can come back and answer that question about the life cycle. Grizwachu, hello. What is the life cycle of a diatom like? So typically diatoms have systematic asexual reproduction where they split and then they keep splitting and splitting and splitting. But as I mentioned a little bit earlier in the stream, they form one valve inside of another valve, which means when they split, they get a little bit smaller. And so what you end up with is each one of the daughter cells that are produced in the binary fission, one of them is a little bit smaller than the other. And as it continues, part of the population stays the same size and part of the population constantly gets a little bit smaller. So each cohort basically has one fraction that's getting smaller and smaller. And as the population gets smaller and smaller, eventually they need to do something about it because they'll run out of material to put inside the cell and space for it. And so they'll switch at some point when they get to a small enough size fraction to sexual reproduction. And so there's sort of like a two-pronged component to it. One of them is small enough size, and the second component is some sort of environmental cue that allows them to recognize it's time to switch to sexual reproduction. And then the whole population will switch to sexual reproduction. Diatoms will sort of divide themselves into male and female like components. One has gametes and the other has eggs. And the ones that have gametes will pop open, the gametes will swim out, they'll find a mate and impregnate the egg, and then they'll create an oxyspore, which is a specialized structure that grows to the original size of the diatom, like a little egg, but it grows bigger. And then they'll be silicified, and then they'll start replicating asexually again. So their life cycle is basically this sort of multi-staged in a way. Then they also have the capacity to potentially hibernate. So in diatoms and other types of algae, we call that encysting, where they basically will make a specialized structure that will allow them to persist. Pacific plankton is getting her first vaccine. I'm so excited about that. So some of them can actually go into these sort of encysted stages where they will sleep or hibernate until conditions get better. Aulacosira is usually one of these type that can kind of encysst or hibernate and then repopulate from that hibernation component. Let's see, Anakin Luke asked, do I do any coding? Generally, I do not. I have people that will code things if I'm desperate for coding, but in the old days, back when I was younger, I knew how to do QBasic and a little bit of JavaScript. I just don't do it anymore because I don't have time for coding, but I can do some programming in R, for example, if I need to. But I don't develop programs. I usually just use it. So it's a programming language, but mostly package-based. And so I don't have to actually do a lot of the coding. I can borrow what everybody else does for code in most cases. I have a graduate student, a PhD student, who is very good at coding. And when I have things that I desperately need coding done for, I usually just ask him to do it. So he's going to be graduating, I guess, sometime this summer. So at that point, I'll have to find some other victim to help me code things if I need it. But generally speaking, I don't need a lot of coding in my job. So in the basic, what I do, it's not really something that diatomists need to be trained to do very well. Most of my coding is just learning it for fun or because I'm interested in it. I mean, like I can do HTML coding. That's not real coding in most cases. So I don't know. I guess it depends on what I would need it for. Generally, I don't need it, so I don't know it. This is an internal view, again, similar to the one that we saw before. I think it's species, I was calling this species 2. And you can see again, costi, Mantifotoportula, alveolar chambers, and then there's some structures on the valve face here that have domed cribra. So normally, if we were looking at a cyclotella, which is what this is probably most closely related to, it would have this, the Rimaportula, on a costi, but they typically do not have areolae on the valve face. So that combination is like, this thing doesn't really belong where people had been sticking it, which is cyclotella. The problem is, we don't have a genus where it has the characteristics of cyclotella, but then has areolae on the valve face. We have ones that are similar, where the Rimaportula is on the valve face, and then areolae are on the valve face. But they don't have spines, and these ones do have spines. So there's some zone here where we have a genus, or incipient genus, that should probably be broken out. That's between ones that we have described and ones that we have here. This is not, strictly speaking, a cyclotella, and it's also, strictly speaking, not a Lindavia, or even Phasanorbis, which is the genus that I erected. I don't think it has the right characteristics for any of them. So, but probably most closely related to Phasanorbis, which is, kind of neat. But I am going to take this picture, and then I'm going to come back and see what people are chatting about. Fubear. This is fascinating. Thank you for the explanation of life cycle. Is the data acquisition completely manual, or are you doing so more automated surveys? Everything I'm doing is completely manual in this, other than, I'm not doing anything while it actually collects the image. But in this case, our data acquisition, like actually collecting the data, is done by me, a person, running the SEM, manually selecting which things I want to look at, manually selecting the focal heights and the composition of the images. So you're seeing me do that right in real time, although I don't have a camera pointed at my hand so you can see what I'm doing. But there's a keyboard in front of me and a mouse and a little digital pad that's associated with the SEM that allows me to control it. So, time for a new genus. Yeah, probably it is. It's a problematic group, and it's challenging to figure out where things belong. In some cases, our decisions, like what we do with where we classify things and what we use to describe or separate them, is sometimes challenging to sort out. So a lot of times we use this characteristic, the number and shape of the strutted processes and their frequency on costi. So you can see there's 2 costi between each one, 2 costi 1, 2 costi 1, but here there's only 1 costi, and here there's 2, here there's 1, here there's 1, and then there's a Rimaportula, which is not one of these structures, but a different one. And then there is one here. So there's two costi, but this one has a Rimaportula between them. And then for the most part there's 2, but this one only has 1 between them as well. So it gets very difficult sometimes to figure out, like, how do you, like, what components should you use, because they're a little bit plastic, right? There's some variability in the living organisms. They're not exactly like limbs or something where you can say, oh, this has 6 legs. Oh, you can't see a mouse while the image is being taken. I guess maybe if I were on this screen you could. Possibly not. But so between each one of the mantle Photoportula, there's a costi. So this is, I think I'm calling this one species 2. Can't see what that says. Oh, yeah. Probably just because I had it in the wrong place. So the classification you use is based on phenotype, which I understand given that you work with skeletons. Is there any DNA-based classification of diatoms? There are. In fact, people have started characterizing diatoms based on their DNA to the point where we have enough of it figured out. I mean, the problem is that not every diatom, and in most cases, most diatoms don't have the genetic information known yet. So we have a number of them where people have, they've got the entire genetic code for them sorted out. But there are potentially millions of diatoms. Species, I mean, not just number. There's like probably 1 million in one of these samples. But in terms of species, I mean, estimates are very high for the number of diatoms. So somewhere between 1 and 2 million is a number that people wouldn't be surprised by if there were that many total number of diatom species. And so that's a lot of DNA analysis to get it to that point. But they do have a good enough handle on them that they've started using diatom DNA to analyze things. So if you wanted to, for example, analyze a river system and see which species are in it, they do have enough DNA information to get some of those species out. Most of it probably we would only be able to sort out at the genus level, though. So not necessarily super useful. They do the metabar coding analysis for some of these sites to try to get a quick handle on things like diversity, and there are for sure people working on it. My approach is largely looking at assemblage information, looking at living organisms, and then trying to figure out, or dead organisms, and trying to figure out which groups of them being found together tell us something about that information, about that site, that would be useful for reconstructing past environments or whatever, water quality. That's a valve face of an Alveolophora, so probably a good thing to have. But there are people looking at the DNA. It's just that it's still quite a ways behind where we are. And there aren't that many diatomists in the world. So on the biology side or on the paleolimnology side. And so, the sparseness of the workers means that there's a lot of taxonomic resolution that's not very well known. And I have at least some colleagues who would argue that we need to stop sort of separating things up to species until we can at least figure out whether or not we're looking at things at the right level in terms of genus. So yes and no. Hello, Table Sick Maniac. How's it going? In any case, even if you had the DNA information, we still use the skeleton to as a foundation, right? So the voucher is the skeleton for the most part in those cases. So we use that characteristics of the skeleton to provide us with most of the information that we need. And then DNA analysis is useful for living stuff, and in some cases, some of the ancient stuff can be analyzed through DNA. But I think in general it's missing a lot still in our field, and I'm not a genetic analysis kind of person, but I work with people who are like that, who will look at the genetic material to try to figure out, what components on the diatom are characteristics that would separate them enough, if that makes sense. So if you're trying to figure out, should this thing be in its own genus, and it's a living organism, a lot of times what we do is rely on the DNA analysis to tell us whether or not it's monophyletic or paraphyletic. So again, that's not something I do. Somebody else does that sort of thing. But we look at the combination of characters and then usually the genetic analysis to try to see how different things are and whether or not that qualifies as something that should be lumped out at the genus level. There's a lot of questions left in diatom taxonomy that aren't really addressed. And, part of that's there's scientific workers, and part of that's that the funding is not, it's difficult to come by for just diatom taxonomy. So we do a lot of our work in my field. The taxonomy is done kind of on the side. Like I do paleolimnology, paleoecology, and then I find new species and describe them, right? So, but I didn't get a grant to describe a species or anything like that. So it's mostly unfunded work. And I don't know if that's necessarily exclusively true, but it's probably where a lot of the taxonomy is done, because it's hard to just get like money to do taxonomy for some tiny organism that most people don't know anything about. This is an Alveolophora, which is a type of diatom. And this is the valve view, the valve face of the diatom. We call the valve view, where we can see the valve face. And normally these things land on their side because they're shaped like beer cans or hockey pucks. And so it's a little bit more common to see them from profile. But the valve face in some cases will tell us useful information. So I just went ahead and collected that image because of that. It's a good question about the phenotype, though, and I think it's really relevant. When you can get that kind of information, it can be highly impactful. This is an internal view. Something very small. I think it's a cyclotella, but I can't even see any of the inside of the diatom because of this huge lip around the outside edge. But I guess it could also be an Alveolophora that we're looking at, and that could be the ring least. But you don't usually see like an internal view of Alveolophora like this, so. Where you can see the valve face from the inside. So I am going to go ahead and take that as well, as long as I'm collecting images. Why not? Yeah, sure. Anytime. IHVH 1969. Part of what I do here is answer questions. And I like it when there's interesting questions where we can think about it a little or talk about it a little. So, thanks for the question, actually. Can't believe I've been on an hour already. That's one of the things that I think is always the case with the SEM. You start on it, and then it just feels like it's just over before you even got anything done sometimes. So we've been on one stub looking at one sample. I've collected maybe 8 photos. DDD Studio, I'm impressed with the resolution of this SEM. You must keep it in very good shape. Thank you, DDD Studio. I work very hard at keeping my SEM very clean. Generally speaking, I force everybody to use gloves when they put samples in and out. And if it's an organic sample that's gone into the machine, I always force people to take those out and put something else in there when we pump it down in between uses. So if it has wet material, plant material, something organic, generally keep that out of the SEM. And the maintenance guy came through about a month ago and did some primary maintenance on the SEM. So he just cleaned out the wine hole cap and realigned everything. But the resolution on our machine is very good. And for tungsten SEM, I feel like we do have very good quality images that come out. And I've had a lot of my colleagues, other diatomists, that see my images and tell me they wish they had my SEM. So, which I usually tell them they can have access to it whenever they like. These are some materials, for example, that colleagues sent me, and I'll run the analysis on it, and then I'll incorporate it into my stream, and then I will, oftentimes just work with them, collaborate with them to publish it. So, yeah. So, and also I spent, I spent maybe 2 months when I first got the machine. Before, I was just using it all summer, one summer, before anybody else really got to use it, after it had been set up. So it was set up 2 years ago in the summer, almost 3 years ago now, I think. And then it was just me using it. And I played with all the settings until I figured out like how to get the image out of it that I wanted. And for diatom stuff, which is what we're looking at here, I usually can get very nice images from it. Very high quality, high resolution, but it's from practice, basically. From knowing what to tweak and what to change. And we're also kind of close. So, and we're on 30 kilovolts. So I'm kind of on the high end. Maybe a little excessively high with respect to the accelerating voltage. And for diatoms, you don't really need to have it that high. I just have it there because it's easier for me to use if I want to zoom in and look at something really small. And I don't like jumping around between accelerating voltages very much. So I like to just leave it at 1 and not futz with it, basically. And I just rotated that a little out of our field of view. But it's a good machine, and I think we've made a good decision in purchasing this particular one. This is one of my favorite parts, which is basically I can rotate stuff without having to reposition it. It just does the math calculations and puts it back in the field of view for me. So, and you probably, well, you didn't get a chance to see it because before we started, for about 30 minutes before we started, I was futzing with the stigmation and playing with the wobble and the column centering and stuff like that. So I like to get it done before the stream happens so that I don't have awkward 5 minutes of me trying to get something stigmated perfectly. So a lot of the quality of the image is a result of that. And I still feel like the stigmation could be tweaked a little bit to be a little bit better than it is, but I always feel that way. I mean, I could get the stigmation almost perfect and still be like, could be better. So this is an Oligocyra. This is kind of an interesting view of it. These are two sibling valves. So this is valve from one diatom and the valve from another diatom, and they live in a colony. and they're interfingered by these spine structures that you can see in the middle here. These things. You can see there's one from this side and one from this side, and then in between one from this side. And these are interesting spines. I would refer to them as, I think they're linking spines because they look like they would be very difficult to separate. And of course, the sample's 6 million years old, so they haven't separated by this point. I don't think they're coming apart. They're being held together pretty tightly. So, and you can see that here. So the spines themselves are actually kind of tumescent. I don't know how to explain it any better, but they're wider in the middle than they are at the tip and at the base. And that's going to make them very difficult to separate. So I think that's why I would call them linking spines. But Alveolophora typically have two different types of spines they form. When they want the colony to stay together, they use these linking spines. And when the colony gets too long, say for example they're trying to become more buoyant, and they want the colony to be a little bit shorter, they will create valves with separation spines that usually causes the colony to separate. And then you end up with two colonies instead of one. But they're shorter. So, I think this is an okay image. You have an FEI inspect, yeah. You're a failure analyst engineer. That's pretty cool. Hey, Mama Bonbon, hello. It's double checks, yes. I'll have to look at some diatoms in the future. Yeah, they're pretty interesting microstructure, ultrastructures associated with them. When we were looking for, so the inspect is I don't remember the names of them. That's a field emission, I'm assuming. FEI got bought out by Thermo Scientific, right? So that's if it's a FEI, it's a little bit older. But we talked to the FEI people quite a bit when we were going to buy this, and the guy used to work for Tescan, which is what this one runs. Oh, it's a tungsten, okay. And they were trying to sell us another SEM, but they used to work for Tescan, and they were like, "Oh, the price you got for that, it's too good. I can't match it." So they backed out, and we went ahead with the Tescan. But what I really like about it is the rotation. We can tilt and rotate the stage in any direction, and it will just recalculate what that position is and move us to the right place so that you don't lose the specimen. And in the old days, I had an old JOEL that I used, that I learned on a Joel, and I had to do it all by hand. So you'd rotate it, and then you'd have to X and Y to get it back to the position you needed to, and then you'd rotate it, and so you could only turn it a little bit at a time. And it'd take an hour to turn a specimen around. And in this SEM, I can just click a button and say, I want it to turn 180 degrees, and it doesn't matter where it is on the carousel. You can see below me there's we're on a 7-stage carousel. But we're on the outside margin, and regardless of the fact that I turn it, it still leaves us in the center. So I think it's really cool. And it's allowed us to do some things like photogrammetry, which I think would be really challenging on a normal microscope to try to get it to turn and stay the same distance. And on a normal SEM, that would be incredibly challenging. So I like that about this one a lot. Right now we're at 28,000 times. So we can get a lot cleaner images if I lower the beam intensity and take a longer image. But for a 3-minute image, I think this is good enough. It gets us where we want to go. And so I think this is the second Alveolophora species. I don't think it's the same one as the first. The spines look very different, but it could be that it's the same and that this is just the linking spine version, and that one was the separation spine version. I don't know. So But yeah, it's nice to have an SEM regardless of uh, what the features are. Um, when I first started here, and for the longest time, I never had an SEM or access to an SEM without spending a truckload of money. And it typically costs something like $50 to $100 an hour to use an SEM if you're in somebody else's lab. And um, and I usually don't have $50 or $100 per hour to do that. So, from a project, because again, they're typically funded. The taxonomy is funded separate from the actual like science. So, all right, I'm gonna hop over to stage 2, and this is the same sample, but it's just a different specimen from it, a different aliquot, and see if there's anything else here, because I feel like we saw most of the centric diatoms in that sample. I think there was 3 different ones. 2 of them were clearly cyclotella. One of them may have been a stephanodiscus or something in between. I just want to see if there's any other views of the... It would be nice to have a nice external view of one of those bigger cyclotellas, cyclotelloids. Gets sort of interesting combination of people who watch the stream. Sometimes they're like you, kind of engineers who are interested in just seeing the SEM, and sometimes they're fellow diatomists that are looking at science, and then sometimes it's just a cross-section of people who are interested in looking at cool things. So there's like a diatom sitting on the surface of this. It's an Alveolophora. So, but it's fun for me to have interactions because I sort of cross all those boundaries as somebody who likes to operate the SEM and sort of be SEM wonk. And then also I'm a diatomist and scientist by profession, and a photographer. So people who are just interested in the images that come out of it are also fun to talk with about the detail you can get. So I hope you're doing well, Mama Bonbon. I don't think I saw you at Pacific Plankton stream on Monday. Had to run everything by myself. And, once you started joining in as a regular moderator there, I feel like, then I got lazy. I was like, oh, Mama Bonbon can handle it. Now look at me. I'm spoiled. Yeah, not seeing a whole lot. So I'm going to try jumping over to one of these other stubs. We'll look at 3, see what's here. A lot of it is very small. I see a diatom here that's also very small, hiding under some ash. Looks like another Alveolophora. And it's got junk in the inside. So that's not a very exciting view. Can't see any of the structure. One of the problems with these samples is that there's so much ash on them, even after we did this heavy liquid separation, that in order to get enough diatoms to see anything, we kind of have to put a lot of material on, and then most of it isn't diatoms. Most of it's like fragments of ash. So it's like a confusing landscape of other things that aren't diatoms. And then the diatoms that are on here are very small. So generally speaking, there aren't a lot of like big, easy to spot diatoms on the slides. So there's a lot of searching. And sometimes that's the way it is. Sometimes we're on a hunt in the SEM. A lot of time is spent just trying to like locate what you're trying to get images of, and then I think most of the time you end up finding something. It's got junk on it or clay on it, or it's not at a good angle. So the cases where we have nice clean shots of things are really rare and pleasant when they occur. Sometimes I just suck it up and take pictures of stuff with dirt on it because you have to. So it's a piece of something. As I mentioned, I was expecting to have a student and another colleague of mine join me for today's stream to help with sort of chit-chatting about what they're working on or whatever. And one of them needed to get vaccinated and got an opportunity to do that, and the other one got sick from something, not COVID, as far as I know. Unlikely, because I think they were also vaccinated. So if you're out there and you can, you should hopefully get yourself vaccinated so that we can move past this current crisis that we're in. I am, as of tomorrow, 100% ramped up, completely vaccinated from both shots. So it doesn't take long, and it's the only way we're going to ever get back to anything normal. So do your part, I guess, if you can. So many colleagues in other countries that I've talked with, and they still have no chance of even getting vaccinated for another 4 months or something. So here at least we have some places where you can get access to it. And I know Pacific Plankton said she was also in the process of getting vaccinated. So happy about that. So we were doing our best to try to help her find a place to get access to the vaccine. She lives in San Francisco, which is like one of the highest demand areas in the US. Not seeing a lot of anything in this sample. Diatom-wise, pieces of some Alveolophora. Let's try this one. Maybe a little bit more promising. There's at least some diatoms in here. Here's something. That is the valve face of an Alveolophora, I believe, which is closely related to Alveolophora. And these samples actually look they're in pretty good shape. Here's the Alveolophora in girdle view. I'm not sure what this big thing is. Maybe an Ellerbachia piece, but maybe it's just a fragment. That is an Alveolophora fragment, or valve face view. There's a lot of more diatoms in this sample anyway. This is an Alveolophora, but the girdle band is still on it, which makes it really hard to see the structure. Oftentimes the girdle bands will fall off of Alveolophora pretty early. And it's impressive because I think this sample is about 10 million years old. So a 10 million year old diatom, and you can still see the most fine structures that it has on it. Here you can see some more spines between, I think these are fragilarioids. Those are not Alveolophora. but you can see the spines here interlocking again. These have linking spines. So they only have linking spines, such that we just call them spines for this group. But you can see how they're sort of woven together to make it so that the diatoms always stay attached in the colony. You can see girdle bands on this as well, still preserved. Again, they're probably the most delicate structure on the diatoms, on these ones anyway. And you can still see the girdle bands attached between the valves. So, kind of cool. The samples are 10 million year old, still just pieces of them still visible. Yeah, I did a stream about photogrammetry from the SEM. We took about 70 photos of one diatom, and we made these three-dimensional models from it. So that was a 3D model that is exclusively made from SEM images. So it's two half models that are put together. It's the same diatom. So we just imaged one of them, took the 70 pictures, and then put them into Agisoft. And I have a colleague who does photogrammetry. They basically turned that into a 3D model, and then we printed it on our 3D printer. So you can have a diatom that you could hold in your hand. This diatom is a species that I also described. So this is Stephanodiscus coruscus. It's from June Lake. And it's an endemic species, or we think it's an endemic species. So we haven't found it anywhere else yet. But pretty cool, and also I think going to be the subject of a couple of papers that I'm working on as well. Still working on your first shot. Yeah, good luck. North Carolina just opened up vaccinations to any adults. Yeah, our university just started taking appointments. They're going to open up a station on campus, which I'm excited about because I really want the students to get through this, so we can start not having to teach in masks and do remote stuff, and have larger classrooms. So Twitch is a great hub for all kinds of things. Yeah, I like it. I like streaming here. I only stream from the SEM about twice a week, and I used to do some light microscope streaming from home. I'm in the process of getting my new microscope set up at home, so it'll probably be another couple of weeks before I do any more home streams also. But in 3 weeks, our semester is basically over, and then I'll probably do a lot more. I'll have a lot more spare time, and we'll be able to do a little bit more streaming from the light microscope and from the stereo microscope as well. So let's see. Micah asked, how do they reproduce when attached together in a colony? Do they separate first? They do not. So they stay stuck together. And what happens is, because the valve of the next one forms inside, they just start to pull apart, and they create a new valve on the inside with the spines, and they grow with the spines interwoven. And then when they go to separate, instead of separating, they're already linked together as spines. So they basically pull apart until the whole valve is exposed, and then there's the colony. So it's kind of a neat trick. You can sometimes see the previous colony components stuck. You can see like the levels where the girdle bands were wrapped around them at a different elevation. I'll see if I can find an example of that for you. This is Ashfall 17. 16 didn't have a whole lot, but 17 did. And these are, geez, I don't know what the genus is. I'm just going to call them Fragilaria. I don't know that they're actually Fragilaria. I can't see the valve face, so 2 valves linking. Normally I would be able to get it into genus, but none of the structures are visible here, so I don't know what to do with that. I can't see areolae, and I need to see the areolae to get it into genus for this group. and I don't see any. So I don't know, that could be something that doesn't go in a modern genus, or it could be something that if I saw the valve face, I would be like, Starcirella. But I can't tell from where we are what that even is, which is kind of cool. This is the internal view of an Alveolophora. It's got junk in it. It's my technical term for any type of debris, organic or otherwise, that might be on the inside. It's probably clay. It's probably mostly inorganic clay at this point. But it's just in my way, so that's how I classify it. Yeah, this is a sponge spicule, I think. I don't think that's a diatom. Solid silica with a little hole run through the middle. And we keep seeing these things, which are the Alveolophora. Alveolophora have an asymmetrical shape. Here, this one here is like the one we saw, but this one is a Starcirella. And this one is either an Alveolophora or an Alveolophora. I'm not sure which, but those are some interesting connecting pieces between them. So that's the girdle band that we're seeing, and then like peeled back behind it, like peeling paint or cracked paint, is the actual valve. And so again, the girdle bands are wrapped around. It holds the cells together. And at the very beginning of my stream, I was talking about how sometimes you can see the inside and the outside of the diatom at the same time. And this is one of those cases. So it's broken, and a broken chunk right here allows us to see the inside of this diatom, which suggests to me that it's actually Alveolophora, not Alveolophora. And then here's the valve face and the girdle band, and then you can actually see a bit of the skeleton. If they had just chipped off a bit more of the girdle band right here, it would have been amazing, because we would have had all the different potential views in one stop. But you can see inside and outside, and then in this case we're looking at the girdle band wrapped around, and then here's the actual valve. And you can see that there's sort of two layers here as well. So I am going to get that picture just so that we can see. This is kind of a neat component just because it has all the structures. It's not very pretty, but in a way it's sort of fascinating because you can see all the different levels, and the inside, the outside, the ring least, which is the structure right here. So like if you were trying to characterize the species, we could do most of it from this one image. We would then need to have it without the girdle band so we could actually see the valve, and then a view of the valve face, and we'd have everything we needed basically. That's a nice pretty image, a really high resolution quality you can see up in here. The valve face and the girdle band. Yep, exactly. The colony just grows. They add a new segment inside, and the parts that were attached stay attached. And so if you're wondering, like for Alveolophora, I was saying they have an option to make linking spines versus separation spines. And if the colony needs to separate, the cell that's made inside of the existing cell, so it could be a cell that normally is linked with its neighbor, and then when the new cell is formed, because half of one of the valve goes to the parent, and half of one of the valve goes to the other parent, right? The two daughter cells that are generated, however you want to think about it, they each get half of what happens from they get to keep half of the parent. So you can have a valve where one end of the valve is a linking spine and the other end is a separation spine, right? And that would be the one where the separation actually would happen in that system. And so the separation spines usually get separated, but you could see a whole colony then where the separation spines would be together. So it just makes a separation spine. And basically it's the only way it could work, right? Because if they separated, how would they ever get back together? So they actually form with the spines interwoven, or if they're separation spines, where the spines fit into a furrow on the opposite valve. And, the structure needs to be that way, because otherwise how would they ever fit the right spine into the right furrow on another valve? everything's made out of solid silica. So, um, they would have to match up perfectly with something, which they don't always have the same number of spines. They don't have the same sizes. So that creates a problem when you're trying to like separate and rejoin. From this image, you can see in here the girdle bands. There's one girdle band here and another one here separated. There's the valve face. And then this is the ring least, which is sort of the internal ring structure on this diatom. And you can actually see into the valve here. So this is what the inside of the skeleton would look like, where the cellular material would normally have been. And this one's closed, so it's not broken in any way, but the girdle bands wrap. You can see there's a little sort of dividing line right here. And then you can see that the girdle band is also wrapped around right here. And these girdle bands have like a little interesting interfingering with their neighbor girdle band right there. You can kind of see that structure. It will be a little bit easier if I were to put it into Photoshop and highlight the contrast. There's a little bit of a overlapping gap right there. So that's one band, and it goes not all the way around, but most of the way around. So kind of cool to see. This is Alveolophora, inside and out. Okay, it's a nice, interesting view of that diatom. And this is, as I mentioned, internal view of Alveolophora, but it's got junk inside. So we're not going to stick with that. Here's another one. Sometimes the three-dimensional nature of diatoms is kind of hard to get your head around because they're small and you can't hold them in your hand very easily. I mean, you can hold a lot of them in your hand, but you can't see them, right? You can't manipulate them, which is one of the nice things about making a 3D model, is that it actually gives us the capacity to potentially explain this stuff a little bit more clearly. Unfortunately, I don't have a good Alveolophora model in front of me. We were doing 3D modeling from SEM images for when we first got it. We tried it a couple of times, and then I don't know. I guess I got too busy with other things and haven't gotten back to it. I was having a student help me with it when we were doing it. I think it was one of the things Mallory was helping me with, and so that's actually really kind of cool. So this is Punctus Striata, it looks like. An ancient Punctus Striata. Could very well be a new species. I think people would have mistaken this in the past for a Starocyrella, but that is for sure not. It's got connecting pieces between them. You can't see this in the light microscope, by the way. You could see the diatom, you can't see the surface structure. And so you would just look at this and see like big openings, and it looks like Starocyrella leptostaron. It looks like an existing species, but I don't think that that is what that is. I think that is something different. And this crossbar that's running through the middle of this suggests that it's not a Starocyrella at all, but more closely related to Punctus Striata. Which is cool. I'm going to take a picture. I'm going to show that to Mark and see what he says. What happens when some of the colony dies, or they just drag the rest of it down with them? If part of the colony dies, I mean, they're phytoplankton, so they probably are under similar environmental conditions, but if like the if like one of the cells dies, it still stays suspended. They don't fall out of the colony. They stay, they're just like a spacer at that point. They're not contributing anything to the production of the colony, but the diatom could create separation spines, and the colony could break apart. Theoretically, they could cause that particle to settle out if it got separated. But I don't think they care. I think the little extra weight associated with it usually gets lost. It's a non-producing component of the colony that's just sitting there. So I don't think it's problematic for them. Is focused ion beam sectioning a diatom thing? I don't know. I don't know what focused ion beams. I mean, you're talking about like laser diffraction. Like, I mean, what do they call that? Where you burn a hole in something and then look at the composition of it. For diatoms, we don't care about composition usually. So it's silica skeleton. Everything's made out of silica. So we don't focus too much on the composition. I could be talking about the wrong thing. But a lot of times, I think what you're talking about is they like focus a beam on one little area and burn a hole in something. People have used a atomic force microscope to look at diatoms, and you can build the whole 3D image of them from those. I have a friend who has a atomic force microscope, but I don't know that he really even uses it that much. I mean, they got it donated to their university, and so he just has it. Hey, Pacific, did you get stuck in the arm? There are dual beam microscopes where you not only have an electron column but also an ion column. You can see all these structures. No, I don't know anything about that. So, subscription. Thank you, Pacific Plankton. Also, my subscription for your channel is about to run out, and so tomorrow, when Pacific Plankton streams, I should be getting mine upgraded as well, or updated. I don't know what this is. I'm going to call it Punctus Striata. I don't even know what genus something goes in. Punctus Striata. I think it's like a Punctus Striata. Like? That's what I'm calling it. Punctus Striata like. I don't know where it goes. I'm going to show it to Mark and see what he says, because it's not a Leptostaron, which is what I thought it was from the beginning. And I guess I've been, let's see, do this, I do that, there you go, Specific Plankton's information, and shout out to her. Also, before I started streaming, I was watching Miss Daisy D. She's got her stream going. I don't know if it's still going, but it was. It's the Luna Moth Emergence Day. So the Luna Moths that she has, like domestic Luna Moths that she raises, they are emerging from their cocoons on her stream today. So depending on what happens here at the end of my stream, we may go there. Here you can see may go there for a raid. We could raid a butterfly. Oh, I guess they're Luna Moths. These ones are little triangular shapes. These are linking spines for sure. But we're in a different slide, so I don't know what the Alveolophora and whatever else are in these samples. But again, you can see this is the ring least. A broken piece of the valve is exposed the inside of the diatom. So you've got a little bit of time before I think it's 2 weeks if is it a Pfizer shot? Or did you get the Moderna? This is a cool diatom. This is Tetracyclus. I was looking for a nice image of Tetracyclus. This one is not a nice image, but it is an image. And you can see up here, this is the Rimaportula. We saw Rimaportula on the cyclotelloids on the other slide. This is a huge one. I mean, that's a monster-sized Rimaportula. That thing is like 2 microns wide. I don't think I've ever seen one that big, except for on some of the really weird Actinocyclus that are ancient. It's a really big one. I'm going to rotate that a little bit. I think I want 140. Pretty good guess for corner-to-corner action. I need to go back and refocus. There we go. Very nice. This diatom's 10 million years old, and all of the structures are still here. And if you're wondering how is that possible, it got buried in ash, and ash has a really high silica content, and what that means is that the groundwater, which might normally dissolve diatoms after millions of years, is supersaturated with silica, and as a result it has a very low probability of dissolving the diatoms at all, because there's a bunch of ash in it basically that's already dissolved into the water. Which allows the diatoms to be preserved to this really pristine state, which is amazing. And some of the best diatomites are like this. They're in ash deposits. So this is the genus Tetracyclus. It has these thick costae that run across the valve, and it is an aeraphid pinnate. And so like the Fragilarias that we were looking at, sort of in that group, although they're off on their own. And these ones have these enormous costae that run across the valves, and then these are the striae. These little rows are striae, and they meet together in a sterna. So the sternum basically runs all the way through the middle axis of the midline of the diatom, and then this is the Rimaportula, and it has one end of the valve with a Rimaportula. So that's pretty common for Tetracyclus and for the Tabulariacea that these fit into. Or they just have one Rimaportula on one end. So this is a really spectacular view of it, and I am going to collect that image so I can also then see what's going on in chat. P-Chops! Thank you for the subscription. Oh, I've got a bunch of subscriptions today. I want to thank everybody. Yeah. Miss Daisy D, if we could maybe give her a shout out if you haven't done that already. Pfizer. Yeah, good. 3 weeks. Excellent. I like it when you get one, then you automatically get the second one. So that's the best. I'm excited for you. So hopefully also no ill effects. I think I got a headache from it, and that was basically it. So diatoms are luxurious. You know what's really cool is P-Chops. You should also give him a shout out. If you can, please. P-Chops is a streamer who plays really cool sort of psychedelic music, kind of sort of ambient sounding music, and a whole bunch of like video crunch sort of stuff that he puts over top of it. He's got purple chickens. He's got a submarine lair, and he's got a giant company picnic planned for millions of dollars. Company picnic, very luxurious. So you can go there. He streams, and then he'll wipe your brain for free. So you can come out with completely smooth brain, which I think is great. Gets rid of all of that stress, right? Lurking while you do some oceanography. Well, I mean, you're in an underwater lair, so it's a good place for it. Hey, the Tetracyclus guide worked. Hypersonic Nerd. Hey man, I was watching a YouTube video about single-celled organisms. Some of them have literally a little... Some of them literally have legs. How can you be single-celled but still have 2 legs, mouth, and hair? I don't think you can. I don't think you can have legs. You can have pseudopods. You're working on phytoplankton identification guide website. Oh, very cool. Marine. Oh, image with the flow cytobot. Yeah, the little hairs are cilia. Those are common in single-celled organisms, including, you know, some things like ciliates. You know, there's different components of a cell, so that's all I can say about that. These are diatoms. They're single-celled organisms, and they can make a siliceous skeleton. They also have internal parts that are a little different. You know, components of the internal, like chloroplasts, are independent. It's a single-celled organism, though. It just has one nucleus for all that information. And that's basically what the important aspect of it is. So this is Tetracyclus. The how it happens, I don't know. It's a long, challenging question about endosymbiotic and endosymbiotic systems that is a little bit more complicated than I can really talk about in a stream, or really understand that well. So I'm afraid I'm not much help on the how. But you can have specialized components of a cell, so you don't need to be... Single cell doesn't mean like all one piece of material. It just means you have one nucleus, right? That represents that entire cell. So diatoms can have, you know, sort of protoplasm they squeeze out through holes, and they have a skeleton, and they're still single-celled. So it's not exclusively like, it doesn't mean there's just like one aspect to it. It just means that it just has one nucleus for that, all that information. There are some surprisingly large diatoms that are single-celled. On Pacific Plankton Stream, we saw some that were where they are like up to 3 millimeters or something like that. It was enormous. A single-celled organism that was, you know, millimeters in size. And there's those things that they called fairy cups or something like that. I think they're single cells. They're enormous as well. So nature's weird and cool and one of my favorite things. So, you know, there's a lot of diverse aspects to it that are interesting. I think that is a chrysophyte cyst, a very boring one. Stomatocyst that's just like a simple round ball. And, oh, while I'm here, that is a phytolith right there. I think that is a saddle, what they call a saddle phytolith. My postdoc, Dr. Chad Yost, works on phytoliths. He's 2 doors down in his own lab now here. And he works with phytoliths, so I've learned a little bit about him. This is a cool one, so I am going to take a picture of it. I almost overlooked it, which is, honestly, after years of looking at diatoms, it's hard for me to see phytoliths. Like, I've got a blind spot where I'm just like, it's not a diatom, it's just junk in my way, and so it's some random shape to me, but these aren't random. This is actually the This is like a structure inside a plant cell, and the phytolith is silica polymerization of that plant cell. So it's like the space between cells usually. And they create these to add a little bit of extra rigidity to their plant's structure. So, kind of cool. Let's see. I'm guessing the real question would have been, what is a nucleus? Yeah. Honestly, I start by getting rid of all the biological material. The nucleus is the central part of the cell. You know, it contains things like the chromosomes, DNA, and it's required for eukaryotes. I can play a biologist a little bit, but generally speaking, I'm trained as a geologist. And so if it's not about diatom ecology or just ecological principles, I'm pretty, like when you get to physiology, I'm usually at a loss, because I wasn't trained in those fields, or those aspects of biology other than entry-level biology classes. So, you know, if it's taxonomy, if it's based on skeletal components, if it's based on diatoms, I probably can answer it. You start looking at a bunch of other organisms, my understanding's probably not much better than most people's. If it deals with evolution, I probably could answer it, as long as it's not about genetics. Yeah, membrane-bound organelle found in eukaryote cells is where the chromosomes are located, basically. If it doesn't have a membrane, then it's a prokaryote. You can Google. I've got one mod who can Google, so there's that. This is an ancient phytolith, a 10 million year old phytolith. Phytolith? I think it's a saddle. The shapes of the phytoliths are how they define them, and I don't think that that is a bilobate, which is the one that I usually see, which looks like a dog bone. So, that's a diatom. This is a valve-face view of that same diatom that we've been looking at regularly in here. Here's this other view, the bottom end view of it. Same diatom. There's one in here hiding, a little acanthes, and this is a diatom as well. This is a pinnularia. If we got really close, which we will, you can see the pinnularia striae have these little tiny holes. And on my good days, my normal, when I see these little tiny holes, my normal instinct is to try to fix my stigmation. This could always just be a little bit better than it is. I don't know that you would even notice that difference, but I do. It looks a little bit better to me. But that's a pinnularia. So Pinularia have a sort of chamber-like structures on the inside. I don't think you can see any of the chambers here. But that's the raphe that runs down the middle of the valve. Not sure what's going on here, actually. It's broken, and maybe this one is in the process of replicating. Somebody asked me that question earlier. Could we see a diatom in the process of replicating? Because it looks like there's another valve inside this valve. It's broken, and it looks like there's a second valve, and it's facing the same direction. Which means maybe that there was another, either two of them back to back, and we're just seeing through the broken piece, or there was one inside and it broke through both of those. So don't know which it is. Both of them are kind of interesting, though. Not interesting enough for me to stop and take a picture, apparently. But I can always come back. That's one of the nice things about this SEM and my stubs, is it's not going anywhere. I can save the stubs, and I will, and come back to them in a future stream, which we probably will, because Mark was not able to join us today, and neither was Joe. And for whatever reason, so we'll probably just reschedule with them some other time, and that'll be fine. You can see they're in these sort of like weird bands. It's because the aluminum stubs that they're sitting on have been milled, and they're just in little depressions, like micro depressions on the milling, from the milling, I would say. Junk. It's a diatom, but it's filled with junk. I know when we were looking at these samples before, there's some really tiny cyclotella in here somewhere. So that's what I've been looking for, is trying to find one. I keep finding Alveolophora instead, and Alveolophora, and some little Fragilarias. I haven't really actually seen any of the cyclotellas, but I know there's some in here, so they can't hide from me forever. They're just really tiny. That's Alveolophora still. This one is Alveolophora. It's got a really cool egg shape, right? It's not circular, but oval or elliptical. And even though it was in focus, it still could be more in focus. So that's nice. Going to go ahead and take a picture so I can see what's happening in chat, and also I need a picture of this thing, so you can see a little bit of the valve on the side. Let's see the structure. It's got these very coarse lines that run down the edge, and then the areolae are sort of squarish. And this structure is a ring-leased. It's like the inside of the parts of the valve that connect for a single valve together. So the other side has the spines, this side has the ring-leased view. Yeah, I don't think it's actually on that website. Alveolophora is how it's spelled. But they're cousins to the Alveolophora ancient dead cousins. Right. What if it's insane in the membrane? Well, then it would be an insane cell. Something went wandering by. An emote. I don't know why, but the word organelle has always been one of my favorite words. Oh. It's nice to see you, Green Dish Towel. I haven't seen you for a while. Those are animal type cells, yeah. Crab claw. Hey, Mom Bon. How's it going? You may have made of simple cells. Well, single-celled organisms have one cell but are a lot more complex. Sometimes they're more complex. Sometimes they aren't. Some of the single-celled organisms have way more chromosomes than we do. So I guess if you think about it that way, they're more complicated. We have like 13, 26, right? Some of these have like enormous numbers of chromosomes. And that's where all the information is. So in a sense, they could be more complicated for single cells. It was pretty close to the spelling I would have had. For some reason, when I was describing Alveolophora to my students for my Diatom taxonomy class, the first year I taught it, I wanted to talk a little bit about them, and it was really difficult for me to find any kind of publication on Alveolophora. It's just like maybe 5 or 6 papers about them, so they're pretty rare. And then to get SEM images was a challenge. And then I started getting samples that had Alveolophora in them. And I was like, oh, I can just use my own images, which is even better. Ring least view. Okay. So it's almost, it's past 3. And I actually have a ton of things to get done today, so I probably won't go for too much longer. I just want to look around and see if I can find something cool or a couple of cool things to image before we stop. This one's kind of cool. This is Starosira, I believe. There's a lot more in this sample than that one that we stopped in before anyway. That is an Alveolophora in valve view, which is what keeps tricking me. There's an Alveolophora in girdle view. That's an Alveolophora again. Somewhere in these samples there's a cyclotella that I want to image still. When they're very small, they're sort of round shaped, and when they get larger, or when they are larger, they are a little bit more elliptical shaped. And I really haven't seen any of the larger ones in any of these materials yet. I don't know if it's because I just haven't, because they're rare and I haven't seen them, or just unfortunate with the, sometimes they get buried under other materials. So I'm going to do a quick tour of the slide and see if I can find any larger ones. And then I'm going to jump over to the other stub for this sample that we have. and see if I can find any other ones on that. There's something right there that looks kind of cool. Oh, I don't know what that is. Raphid Diatom. I'm not sure what the species is, and I don't even know what the genus is. So it looks like maybe it could belong to Anomalies or something similar. I did see a Brachiocyra earlier, so those are also closely related to Anomalies. More Alveolophora. Tetracyclus. Slightly different shape, but it's the same genus. The other one we had was a pretty good view of it. A nice big chunky Rimaportula pointing towards us. My laptop is telling me it's going to die soon. Oh, that's an interesting view. You can see the pores on the valve face have really complicated internal structures on them. Okay, let's jump over to 6, and that'll be our last stop for today. I'll see if I can find anything exciting in here for us to look at. Looks like it's a little bit more dense, which is a good sign. I'm not sure what that is. It still has the girdle band on it, but that's a tetracyclus there, I think. So before we get to the end of the stream, I should also say thanks again to Mallory for preparing these samples. I haven't seen Mallory on the stream for a while. That is her choice, not by my choice. But I think she's just busy. It's sort of at that point in the semester where everybody's a little bit tired of everything. So I think she's tutoring at this point, though. So there's a little round guy. I think that's still an Oligocyra. Just looking at it, a little shallow valve. Not sure what this is. It's too much junk on it. I can't even see it, but it's a weird shape. Still valve view, still Alveolophora. It's most of what we found in these samples so far today. Still, I think it's Alveolophora. You see this sort of structure on the outside in here, very barely see it. But that structure is Alveolophora's thick, sort of thick costae up against the ring least. There's another one, Alveolophora. I'm just skirting around the outside edge, middle outside edge of the valve here. See if I can find anything good. It's a tetracyclus again, but broken. This is Alveolophora, I think. Another little Alveolophora, a tiny one next to it. There's another one of those phytoliths that we saw, that we imaged. Same sort of weird saddle shape. Potentially something good here. Nope. That is just ash. That tricked me into thinking it was a diatom. I think that's a piece of a sponge. Yeah, that's all one piece of silica. It's a little broken fragment of a sponge spicule. Here's another phytolith. But I saw another diatom up here. I think it's a Nitzia. I'm not sure about that though. Nope. There's a raphe running down the middle of the valve. Some kind of navicula. I think. Can't be sure. That raphe end looks a little weird for navicula though. Also, its general shape looks kind of odd for a navicula. It's slightly longer in one direction on one side. Let's see, 80? Maybe? I'm doing pretty good with my guessing. Maybe it's a gomphonema, because it is pretty asymmetric. see, if it was a Confinema, this end should have an apical pore field, though. And I guess it does. I guess that looks like maybe there's an apical pore field there of some type. So I'm going to treat it like it's a Confinema. It's a little too asymmetrical to be Navicula. It's been a while since I've been back in chat. Hopefully I didn't miss too much. Oh, Devil Mrs. J, hello. Study with Mossy. What are your samples from? These are from Ashfall Ashfall State Park in Nebraska. Let's see. Is your microscope causing damage to your samples? I remember seeing a video about crazy microscopes like that, and it could cause small damage as electrons impact the sample. So, electron beam hits the sample, and then it knocks some electrons out. So yes, it damages the sample, but the damage is so negligible that I would need to be like sitting on the sample for days in order to, like burn a hole in it or something like that. And I'd have to have the SEM beam bright, and I'd have to have it down in a pretty narrow little band in order to do that. So on a very fundamental level, it is damaging it, but it's so minor that I don't think that you would actually think of it as being destructive. I could put the sample back on and re-image the same diatom probably hundreds of times and not see any difference from one time to the next. So it's not destructive in the sense, in the strictest sense, but in the technical sense, yes. School's almost over, final push. Yeah, that's basically how I feel about it. Yeah, on a very small scale, it is. You're right, Micah. And but also in a more general sense, it's not really. So, and thank you for shouting out Devil and Mrs. J, and thanks for hanging out with us here, Devil and Mrs. J. It's nice to see you again. Yeah, and I think it's a Gumphanema. So it has characteristics of Gumphanema. This little hole right here is a stigma, and then it's got 2 branches on the raphe, obviously, and then the areolae actually have sort of a C shape. So that's not. Initially, I thought they were just straight lines, but when I got down here, I realized they were a little more C-shaped, and then it's got a weird, like angular shape to it. It's like that distance is different than that distance. It's not a perspective thing. We're looking at it straight up and down, so it looks like there's a perspective thing, but there's no perspective. So it should look, linear across the middle and not like tapered. So let's see. This is some sort of a Gumphanema, like Gumphanema sarcophagus. Looks like a sarcophagus to me. Some sort of a coffin shape to it in general. And that is the valve face of one of those Alveolophoras. Which I think I will collect an image for, just so that we have one with the valve face. I think we have some from Girdle View from before, but I might also grab one in Girdle View as long as we're here for the last couple of images. I think that is good. It looks like it's in focus to me. Oh. I've got a paper I need to work on and a bit of a proposal to review and some emails to handle, and I still have to put my Dr. Mo lecture together for this week, which my plan right now is that it will be extremophiles, in particular psychrophiles. So a little bit easier than some of my other lectures to construct, probably, because I already know a lot about psychrophiles and the environments that they live in. So but it'll be fun to talk about. Psychrophiles. Yeah, and on Friday, I should mention that as well. On Friday, I have an interview sort of thing with Freckled Science. So Amanda and I are going to have some conversation about what is science and how we regularly deal with failure as scientists. And I think it's supposed to start at 6 p.m. And then I'll have office hours for Dr. Mo at like 9 o'clock on Friday. So Friday will be a big streamy day for me. So it'll be busy for the rest of the week. But my semester's starting to ease up a little bit. I don't have any class in the morning on Thursday at all until like 2 o'clock. So that's a huge opening in time for me. It'll let me get a little bit more work done. And then, like I said, 3 more weeks, semester's over. My afternoon class on Thursdays, I switched us to just watching videos for every Thursday because I felt like the students were kind of getting burned out as well. And we didn't have any spring break, and I still want them to learn things, but I also am okay with them vegging out a little bit. So rather than trying to force them to have conversations that end up feeling a little extra forced because everybody's tired, I thought, oh, we'll just watch some cool videos. And I watched a really interesting video over the weekend about nuclear energy, which was kind of cool because that's what our topic is this week. It was my lecture on Tuesday. And I don't know. Nuclear energy is sort of like an interesting problem for us as a society because it's not a renewable resource. So, it can't be the only solution we come up with if we want to deal with our impending energy crisis. But at the same level, if you look at who has the lowest carbon footprint of any nation in the world, it happens to be France. And people make a lot of noise about how great Germany is and their recycling and their whatever energy approach, but they opted not to use nuclear energy, and France has got like 80% of their energy comes from nuclear energy, and as a result they are one of the cleanest environmental countries in the world. And in fact, actually, they are the cleanest. So, there's pluses and minuses associated with it that are kind of, I don't know, complicated and interesting. But also, I don't think we can move forward without nuclear energy in our playbook. So, as much as people have a history of disliking it from a sort of environmentalist point of view, and it still does take a lot of carbon to mine it and to build the cement structures associated with it. But there's also a a value to it once it's constructed. So that makes it a gateway that we probably need to take. So if we want to get rid of fossil fuels faster, which we should. So this is a girdle view of Alveolophora, and it's actually 2 valves. They're very short, and they have this sort of crenulated spine structure in the middle here that you can see. They kind of fit a little bit like puzzle pieces together. And then the entire colony is curved, so there's like a noodle, right? Like a macaroni noodle or something. It's like curved towards us, so we can actually see the ring-leased valve, ring-leased view of both of the valves that are here, which is kind of interesting. Just laying on the curve. Okay, so I think this will be my last image for today, and I'm going to try to get that just to be a little bit sharper as a result by changing my speed settings to an 8. And that's a 10-minute photo, but we have 5 minutes left before I was going to end anyway. So this will just give me a little bit of chance to say our goodbyes and whatnot. And I do want to adjust for the brightness a little bit. So I will let the instrument figure out what that is. It usually does a pretty good job with brightness. Yeah, I think it'll be good. So I'm going to start that, and I will let that image cook, and I can come back to chat and talk a little bit. Nuclear scares people more than it should. Yeah. Exactly. A 10-minute photo. How about put 1 hour photo out of business here? Yeah, I think that probably has already happened. Yes, Commander Chef Fard, this is an electron microscope, a scanning electron microscope, and you're here right at the end of my stream, which usually starts at 1 o'clock on Wednesdays and Saturdays. And I think I will be streaming on Saturday this week. I have some ideas. I think what I'm going to do is take take a couple of the stubs and put them on my hat, like attach them somehow to my hat, and then I'm going to go for a bike ride. And I'm going to try to ride around the stadium where all the trees are pollinating right now because it's spring. And I think what we'll do is try to collect all the pollen on the stubs by riding around on my bike and see how much pollen I can get in that process. So they should just stick to the carbon tape as I ride my bike around. And I don't know, maybe I'll even stream myself riding my bike around a little bit. Who knows? And then when I come in on Saturday, I think I'll just do like a pollen stream, which would be kind of fun. I might have some backup plans in case there's no pollen on my actual, like if I don't actually collect any, or if it fills up with dust or other junk. So I may also see if I can find us some other things, but I think it will be a non-diatom stream. I think we'll look at other things on Saturday, so that'll be fun. And then maybe we'll do it again for next Wednesday. I don't know what's going to happen at this point. I don't really have a good plan for it, but we'll look at it as we'll see what happens, right? Everything's completely yellow from pollen where you live, yeah. Yeah. We could definitely also collect some stuff that way. I just wanted to try it, like on my bike. Part of the reason I suggested that approach is because, put your noses on high alert. Part of the reason I suggested that approach was because the stadium for campus has planted around it every tree in Indiana. So they have like every tree that's found naturally in the state is planted, at least one of them, around the stadium with a little plaque that tells you what the tree is. And so I thought, oh, if I just ride my bike around there a little, I get some exercise and also I can collect some pollen for the stream. So that was my idea. And I had an idea that I would do it in the spring, maybe a couple of times to try to see if different pollen gets stuck on my SEM stubs. Yeah, they planted them that way. They went around and basically did every one of them with a label. So, yeah, it should be fun. And if I also probably will just collect some pollen. There's a bunch of plants with pollen on them right now, so I can, I've got sticky tape on the stubs, so I can just kind of dab them on the plants and pull up some of the pollen that way. So I think maybe we'll look at a little bit of pollen. It'll be maybe also for Wednesday next week. I don't know, but that's sort of my intention at this point. We'll see how it goes. So, we've had 100 diatom streams in a row, so probably would be reasonable for us to look at something else every once in a while. Yeah, actually, why don't we, I see that Freckled Science is actually on right now, so why don't we raid her, and that'll be a nice connection because we are going to be on her stream on Friday. We're going to talk about failures as scientists together. It's part of one of her new series that she's doing, and I think it's going to go onto YouTube or something like that as well. So I set us up. We'll run over there and we'll check out Freckled Science. If you don't know Amanda, she's a highly energetic streamer who I don't know what she's doing. A lot of times she does 3D printing, and also sometimes she does experiments in her house. But she's a fun character, and I like to come visit her when I'm not streaming, just to sort of see what she's up to. But she's got a good crowd of kids there that we'll interact with on Friday. And you should also check out Pacific Plankton Stream, which is going to be tomorrow night, right? It might not be too high energy if she's in don't talk to me mode. Well, I think she'll still be excited to see us, so. You accidentally glued yourself to an alligator? I need to hear about that. Okay, well, in any case, I want to thank everybody for hanging out with me today, and Pacific Plankton. Oh, somebody did, okay. Micah and Pacific Plankton for helping to moderate all the visitors we've had. OpenSet, it's nice to see you. You should check out OpenSet, and also I've got this squad listing here, but we're getting ready to raid. So these are people you could check out if you're interested. Otherwise, I'll catch you next time.